Primary non-specific defences against pathogens in animals. So there's the skin, which consists of the epidermis and the dermis. The epidermis is a tough outer layer that secretes sebum to waterproof skin, and keratin is also secreted to toughen the skin. It is 20 to 30 cells thick, whereas the dermis is 20 to 40 times thicker than the epidermis. The dermis contains sensory receptor cells, capillaries and hair follicles. The lower layers of the skin are the site of cell division, which replaces cells lost from the surface. Sebaceous glands secrete lactic acids and fatty acids, forms acidic environment and disrupts bacterial processes. There are also mucous membranes, which are the lining of the organs involved in protection and absorption. They are present at most interfaces between body and external environment. They secrete sticky mucus and lysosome enzymes and can be lined with cilia. There is also blood clotting and skin repair. The body prevents excess blood loss, and clots act as temporary seals. When platelets come into contact with collagen in skin or the wall of damaged blood vessels, they adhere and begin secreting several substances. The two main ones are thromboplastin, an enzyme that triggers a cascade of reactions resulting in the formation of a blood clot, and serotonin, which makes the smooth muscle in the walls of the blood vessels contract, so they narrow and reduce the supply of blood to the area. After the clot, a scab forms and the epidermis heals more permanently. There is also inflammation. Infections, mechanical forces, chemicals and extreme heat or cold can damage tissues, causing the non-specific process of inflammation. The goal of inflammation is to clean up the damage and start the repair process. Inflammation begins when damaged tissues release chemical messengers such as histamine, prostaglandins and leukotrienes. These chemicals cause nearby blood vessels to expand and become leakier, allowing more blood flow to the damaged area. These chemicals also attract white blood cells to the site to remove debris and foreign invaders. The results of these activities are easily observed when the skin is inflamed: swelling, redness, heat and pain. This is caused by damaged cells. Damaged cells release histamines, which causes the capillaries to dilate, increases tissue fluid, increases the flood supply and increased phagocyte supply. Damaged cells release chemokines, chemicals to which can show chemotaxis. They attract phagocytes. Phagocytes can invade the tissues and carry out phagocytosis. There's also coughing and sneezing, reflexes that expel pathogens in trapped mucus, which effectively removes the pathogen from the airway tract. Neutrophils are a type of white blood cell that defend the body against disease. Their flexible shape allows them to engulf foreign particles or pathogens. The many lysosomes in their cytoplasm contain digestive enzymes to break down the engulfed particles. Neutrophils are also a type of phagocyte. They're the first white blood cells to respond to a pathogen inside the body. Neutrophils move towards a wound in response to signals from cytokines. Antigen-presenting cells are created when phagocytes present a pathogen's antigens on the surface cell membrane. This then activates other immune system cells such as the T lymphocyte and the B lymphocyte white blood cells. Stage 5 on the diagram demonstrates antigen presenting on a phagocyte. Roles of molecules in the immune system. Cytokines are proteins that act as messenger molecules which bind to the receptors of neutrophils in order to make them move towards a wound. They're released by the cells at the site of the wound. Opsonins are molecules in the blood that attach to foreign antigens to aid phagocytosis by tagging them so that they're more easily recognised. They work in different ways. Some hide the negative charges on the membrane of the pathogen, making it easier for the negatively charged phagocyte to get closer to the pathogen. There are a number of different opsonins, however, antibodies such as immunoglobulin G and immunoglobulin M have the strongest effect. Phagocytes are types of vesicles that are created to enclose pathogens engulfed by phagocytes. Lysosomes are specialized forms of vesicles that contain hydrolytic enzymes. They're responsible for breaking down pathogens ingested by phagocytic cells. Neutrophils versus macrophages. Neutrophils make up 70% of all white blood cells in the body. They have a lobed nucleus and they're involved in the non-specific defense system only. Macrophages make up 4% of all white blood cells in the body. They have a simple round nucleus and they're involved in both the non-specific system and the specific immune system. Antibodies are Y-shaped glycoproteins called immunoglobulins. They bind to a specific antigen on the pathogen or a toxin that has triggered the immune response. Antibodies are made up of two identical long polypeptide chains called the heavy chains and two much shorter chains called the light chains. These chains are held together by disulfide bridges. There are two regions, the variable region, which is the binding site for antigens, which is different on all antibodies. This creates the specificity of antibodies. And there's the constant region. The rest of the antibody is always the same. When an antibody and antigen bind, it is known as the antigen-antibody complex. How antibodies defend the body. The antibody of the antigen-antibody complex acts as an opsonin, so the complex is easily engulfed and digested by phagocytes. Most pathogens cannot further invade a host cell once they're within an antigen-antibody complex, rendering them ineffective. Antibodies act as agglutinins. This prevents pathogens spreading through the body and enables phagocytes to engulf more pathogens easier. Anti-agglutinins clump pathogens carrying antigen-antibody complexes together. Antibodies act as antitoxins, binding to the toxins produced by the pathogen, making them harmless. T lymphocytes mature in the thymus gland. T lymphocytes include T helper cells, T killer cells, T memory cells, T regulator cells. We'll go through them in more detail here. T helper cells contain CD4 receptors on their cell surface membranes. These bind to the surface of antigens on APCs as mentioned previously. They produce interleukins, a type of cytokine. Interleukins, these stimulate the activity of B cells, which increase antibody production. They also stimulate the production of other T cells and attract and stimulate macrophages to ingest pathogens with antigen-antibody complexes. T killer cells, these destroy the pathogen carrying the antigens and produce a chemical called perforin. Perforin kills pathogens by making holes in its cell membrane, making it freely permeable. T memory cells. These live for a long time and are a part of the immunological memory. If they meet an antigen for the second time, they divide rapidly to form huge numbers of clones of T killer cells that destroy the pathogen. T regulatory cells. These suppress the immune system, acting to control and regulate it. They stop the immune response once a pathogen has been eliminated, and they make sure that the body recognises self-antigens and so does not set up an autoimmune response. Interleukins are also important in stopping this occurring. B lymphocytes mature in the bone marrow. B lymphocytes include plasma cells, B effector cells, and B memory cells. Plasma cells produce antibodies to a particular antigen and release them into circulation. Active plasma cells only live for a few days, but they produce around 2,000 antibodies per second when alive and active. B effector cells. They divide to form the plasma clone cells. B memory cells live for a very long time and produce the immunological memory. They're programmed to remember a specific antigen and enable the body to make a rapid response when a pathogen carrying that antigen enters the body again. This is the end of the specific immune system. Immunity. Natural active immunity is when the body itself has produced its own antibodies and or memory cells. The specific immune system produces T and B memory cells, so if the body encounters the same antigen a second time, it can be immediately destroyed. This is present in normal, healthy mammals. Natural passive immunity is when the baby receives the mother's antibodies either through their placenta or their milk. Some antibodies cross the placenta from the mother to her fetus while the baby's in the uterus. This means that the baby has some immunity from disease. Colostrum is the first milk a mammalian mother makes. It is very high in antibodies. The infant gut allows the antibodies to pass into the bloodstream without being digested. So within a few days of birth, a breastfed baby will have the same level of antibody protection against disease as the mother. This immunity lasts until the baby can produce their own antibodies and so develop their own natural active immunity. Artificial passive immunity is when the antibodies are formed in one individually, usually an animal, then extracted and injected into another individual's bloodstream. This provides temporary immunity and can be used for some potentially fatal diseases. An example of this is tetanus. People can get infected with tetanus through an infected cut. They are then injected with tetanus antibodies which are extracted from a horse's blood. This prevents the development of this disease but does not provide long-term immunity. Artificial active immunity, vaccination. This is when the immune system of the body is stimulated to make its own antibodies via being injected with a safe form of the antigen you want to protect the body against. This is the process. A pathogen causing the disease or illness is obtained and genetically modified to be harmless to the body. This isolated pathogen is then combined with other vaccination components such as stabilizers. This is then injected into the patient's bloodstream. Once the non-self antigens on the pathogens have been identified, the immune system's B lymphocytes produce antibodies to combat the antigens. As the pathogen is not deadly, the immune system easily destroys it. B lymphocytes are then kept as B memory cells for that specific antigen. If that antigen invades the body once more, the person's immune system can easily combat this pathogen effectively, stopping the infection from spreading, by quickly producing pre-established antibodies.